Hello, and welcome to today's installment of the 2020 Tribal Alliance for Pollinators webinar series, Data Collection for Seed Collection. Our presenter today will be Amy Buthod, who is a botanical specialist at the University of Oklahoma, working with the Oklahoma Biological Survey and the Oklahoma Natural Heritage Inventory. She conducts floralistic inventories throughout the state of Oklahoma, maintains the list of rare plant species for the state, and is the collections manager for the Robert Bebb Herbarium at University of Oklahoma. Today's webinar is part of a multi-year project that TAP has undertaken with support from the National Fish and Wildlife Foundation to expand our regional seed bank of native plants. Seeds from the seed bank are available for any tribal entity or tribal citizen to use at no cost, as well as our lending library of equipment that is for supplies and equipment needed to restore native plants. If you're interested in contributing seeds to this effort or withdrawing seeds from the seed bank, please contact me at jane@nativebutterflies.org. After today's presentation, we will take questions at the end, and you can send us your questions by typing them into the question or chat box found in the dashboard on your screen. You will be muted during the presentation. So at this point, I will turn it over to Amy. Welcome, Amy.
Thank you, Jane, for your nice introduction, and thanks to everyone for tuning in today to my webinar titled Data Collection for Seed Collection. To ensure we have the highest quality seed collections, we need, of course, good seed, but we also need good data to go along with that seed. Specifically, we need to know that the seeds you have worked so hard to collect are identified correctly with a Latin or a scientific name, and that they are paired with a scientific specimen so we can confirm that identification. We also need to know where exactly these seeds were collected. This data will make sure that the right seed is planted at the right location or habitat, maximizing the impact of the hard work you've done for the Tribal Alliance for Pollinators. Now, what sorts of data do we need to collect along with our seeds? In this webinar, we will discuss Voucher specimens, field data forms, and briefly using the iNaturalist platform. One of the goals of TAP's National Fish and Wildlife Foundation grant was to create voucher specimens to assist with the training of seed collectors and to improve the accuracy of the TAP seed bank catalog. In most cases, it is impossible to accurately identify seed material alone to species. Seeds are often just too similar in appearance. So we need to learn how to create these so-called voucher specimens. We also need to make sure we have recorded basic data about our voucher collection. Information such as the location where the collection was made, the date on which it was collected, and who did the collecting. We get that data from a field data form that will be filled out as the voucher specimens are collected. We can also use an online platform, in this case, iNaturalist, to help collect our data and to share it online. This is another tool we have at our disposal that will aid in the identification and documentation of our seed collection. Now, what exactly is a voucher specimen? A voucher specimen is simply a representative sample of an organism that has been identified by an expert and deposited in a location, such as an herbarium, where it is available for researchers to examine and to study. To make these voucher specimens, we will be digging up or cutting off pieces of actual plants at our field site, pressing them until they are very flat and very dry using the plant press, and then preparing them in such a way that they will last a very, very long time. These voucher collections will be stored at OU in our herbarium and also at the Tribal Alliance for Pollinators facility. These are some examples of voucher specimens. These are actual real plants. As you can see, they are very flat and they are attached to paper. The paper is a standard size, 11.5 by 16.5 inches, and is pre-printed with our herbarium catalog number up at the top. We do that for tracking purposes. All materials used to prepare these specimens are archival, meaning that they should last for a very, very long time without any deterioration. In fact, the oldest herbarium specimens known today are over 450 years old. Just as an aside, because it's kind of interesting, here are some examples of very, very old voucher specimens. These specimens were collected in the early 1800s. I believe 1810 is the actual date. Rather than individual sheets, as was shown in the previous slide, these specimens are actually bound together in a large book. It's probably about 6 inches thick and maybe, a 1.5 feet wide. It's a really big thing. This is the way it used to be done back in the old days. This volume, along with a few other similar ones, are at OU at our Herbarium. We don't know of their origin, but they're beautiful objects with thick handmade paper, and the ink actually used, someone told me, is made from acorns. So, the Herbarium. I mentioned that our voucher specimens will be stored in an herbarium at OU. What is an herbarium? An herbarium is simply a collection of pressed and dried plant specimens filed in a logical way so they can be easily located and retrieved for use. It's sort of like a plant library. These photos are from the Robert Bebb Herbarium at OU, where I work. The cabinets on the left-hand side of the screen, we have about 200 of those. Each of the cabinets are filled with these file folders, and in the folders you see, those are all voucher specimens. We have approximately 250,000 of these at OU. So why do we collect these voucher specimens? Voucher specimens are crucial for the proper identification of organisms. There are often minute differences used to tell apart various plant species, such as measurements in millimeters or the shape of tiny, tiny hairs. Therefore, it is important to have actual material on hand to which we can refer. This is the main reason we are collecting voucher specimens for TAP. We want to make sure that the seeds we are collecting are properly identified. Voucher specimens can also provide valuable data on topics such as species distribution, evolutionary relationships, and climate change. An herbarium will have collections made throughout time and space. We could see plants collected from all over the world and from many points in time at an herbarium. DNA samples can also often be obtained from these collections. There are other reasons for collecting voucher specimens. For instance, there is the concept of reproducibility. You may remember learning about the scientific method back in school. Reproducibility is part of that. It means that a result obtained in an experiment should be able to be reproduced. For instance, if I do a study on how coneflower species are related, I put voucher specimens of my study subjects in the herbarium. If someone disputes the results of my work, they can go and retrieve those exact same specimens and use them in a new study to see if I was right or wrong. Now that we know what voucher specimens are, we need to know how to collect them and prepare them. What supplies do you need to collect voucher specimens? Well, this is easy, and you probably have this stuff at home already. Basically, you need a digging tool, such as a trowel or a shovel, to get the specimen out of the ground, and a plastic bag to put the specimen in until you can press it. Of course, you will also need a plant press, which is the wooden thing you see on the right side of the photo. Plant snips or gardening shears are also handy if you are collecting large plants. You may have to cut them into pieces to fit into your plant press. So you've gathered your collecting supplies, and now you're ready to go out in your field. You'll probably have some idea of the species you are looking to collect based on lists provided by TAP. And when you find them, you will first need to determine how abundant they are at that particular site. You don't want to collect something that's rare or unusual. Ideally, there should be at least 50 individuals of a particular species present before we would want to collect seeds. So we are looking for populations of approximately that size. Of course, these are just estimates. We're not expecting you to go out and count every stem. Just use common sense and good judgment. Once you have determined that the population is of sufficient size, you will want to select an individual, a single plant, that is representative of the population as a whole. You don't want to get the biggest plant you see or the smallest because it fits better in the press, or the plant with the most flowers because it's the prettiest, or the plant that has been chewed to pieces by insects. Just choose an average individual. It's also crucial that the specimen you select has flowers or fruit. Flowers or fruits are absolutely crucial for the proper identification of a species, and we must have those on your voucher specimen. Flowers are actually preferred over fruit. You also want to try to collect the entire plant, including its roots, which are sometimes used to differentiate species. Dig the plant carefully out of the ground using your trowel or shovel. Take the soil off the roots ever so gently, and then put the plant in your plastic bag. That's it. You've collected a voucher specimen. Obviously, if you are sampling trees or large vines, which I don't think we will be doing with the tap work, you can't collect the whole plant. You can't collect a whole tree and glue it on a sheet of paper. In those cases, a piece of the plant with flowers or fruit, along with leaves or twigs, is sufficient. After you've collected all your specimens at your field site, you will need to press them in a plant press. A plant press is simply some wooden strips nailed together in a lattice pattern that allows for airflow. This will help the specimens to dry. Within the wooden lattice, you have a series of layers of cardboard and newsprint. The plant itself is placed in a piece of folded newsprint, and that newsprint is layered along with a heavy felt-like cardboard that absorbs moisture and a lighter corrugated cardboard that allows for airflow. There's also a set of straps that goes around the entire structure. These are pulled tight to hold the whole thing together. These photos show some of the steps involved in pressing the specimens. You simply arrange your plants inside the folded paper so that there is a layer on top of the plant and a layer below the plant. Those of us who make lots of collections buy unprinted newsprint in bulk, but regular newsprint cut to fit the size of your press, as is shown here, is just fine for you to use. When you're laying out your specimens in the newsprint, make sure crucial structures like flowers and fruit can be seen once the plant has been flattened and dried. You don't want them covered up by a leaf or another part of the plant. They need to be visible to aid in the specimen's identification later on. Finally, make sure you label the outside of your newsprint with some identifying data, such as the date and the location where the specimen was collected. We'll discuss this part in more detail a little later in the presentation. So what do you do if you've collected a big plant like this Canada goldenrod? This is a job for your plant snips or your garden shears. Cut that thing into pieces and press each piece individually. However, don't do this. Do not let any pieces of the specimen stick out of the newsprint. It won't get properly flattened, and we won't be able to mount it on the mounting paper. Each piece of newsprint containing each of your specimens will then be placed between the layers of cardboard. You'll have a layer of the corrugated cardboard, a layer of the felt-like cardboard, and then your newsprint. On top of that, corrugated cardboard, felt-like cardboard, newsprint. Just making layer after layer after layer until you have pressed all of your specimens. The result is sort of a sandwich of cardboard, paper, and plant. After you've made your sandwich, you need to get the whole thing in the plant press. Make sure all your layers are stacked neatly and evenly and you don't have anything hanging out like in that previous slide. And also make sure that the pieces of the press are turned in the right direction with the shorter horizontal pieces on the inside. One piece of the frame goes on the bottom of the cardboard stack. The second goes on top. You place your straps around the whole thing, making sure the fasteners, which are usually some sort of buckle, are facing the same direction. And now for the fun part. Tighten those straps. This is the most effective way of doing it. Stand on them and pull them tight, tight, tight. You look pretty goofy, especially if you're doing it in public, but you want them as tight as possible so your plants dry as flat as possible. I've done this in really strange places, fast food parking lots, had some pretty strange looks. Also, don't fall off your plant press. The next thing you have to do is dry your specimen. You want them to dry as quickly as possible so that they don't mold. At the herbarium, we use a special cabinet that has a heating element built into it, or this contraption that consists of a wooden box that sits over a strip of light bulb. It's kind of scary and most likely a fire hazard, so we try not to use that very often. Since those options are not available to you, you can just use your car. Put the press in your trunk or your back window for a few days. Check on it from time to time to make sure the straps are still tight. They will loosen as your plants dry and shrink in size, but you want to keep them tight as possible so that specimen is as flat as possible. When the plants are finally dry, they will end up looking something like this. As you can see, this is a pretty flat plant. Keep in mind that not all specimens will retain their color like the swamp milkweed in this photo. Whether or not a specimen retains its color can depend on factors such as how quickly it was dried or what species it is. For instance, some species in the broomrate family, that's the group of plants that are related to our paintbrushes. Those plants will dry completely black. This is the last step in the voucher making process that you will most likely be involved in, but there are a few more things that need to be done before our vouchers are ready to go into the herbarium. The specimens you've collected will be bundled up and sent to me at OU, where we will finish preparing them. Once we receive your specimens, we will identify them or confirm the tentative IDs that you've given them. We do that using books called dichotomous keys. Dichotomous keys give you a series of choices about characteristics of the plant that will eventually lead you to the correct scientific name. Again, as I mentioned, a lot of these characteristics are teeny, teeny, tiny. So we also use hand lenses. That's what the person on the left is squinting through, and dissecting scopes to view them. This slide shows the equipment involved in mounting specimens on their paper, although equipment is a little overboard. But basically, all we use is thin Elmer's glue brushed onto a piece of glass. The flattened plant is dipped into the glue, arranged on paper, and weighted down until the glue is dry. The glass plate is there in the foreground, and you can see specimens drying in the background. Those little metal things all over them are the weights we use. And as you've already seen, here is the result, the completed voucher specimen. You might notice that we didn't talk about what's in the bottom right corner of the specimen. Every specimen will have a label in this position, and this label is just as important as the plant itself. Specifically, we must have two important pieces of data for our voucher specimen to be complete. We need the location where the plant was collected and the date on which it was collected. Without these two pieces of information, our specimen just isn't very useful. Specimen labels can include other information too, such as the habitat where the plant grew. Did it grow in a prairie? Did it grow in a woodland? Did you find it in a pond, etc.? Also, we want to know how abundant it was at the site. What soil type did it grow in? What other plants were growing with it? These are all good pieces of data to include on a specimen label. So where does this information come from? Let's go back to our collecting supplies slide. In addition to the things we need to physically get the plant out of the ground, we also need some things to collect data about the plant. For our work with TAP, we're going to use a standardized field data sheet and cell phones to help us collect our data. This is our field data form that we have designed. We tried to make it very quick and easy to fill out in the field. There is a space for your name, the date, a tentative identification, location information, and habitat information. For each specimen you collect, there will be a corresponding field form. This is the information you could use on your newsprint in an abbreviated form when you are pressing your specimen. Under location data section, you can see that you are also asked to include latitude and longitude. You may be wondering where to get that information. That's where your cell phone comes in. You will need a mapping application of some sort downloaded onto your phone. In this example, I've used Google Maps. For Google Maps, your location will appear as a blue dot. You can tap this blue dot and a red pen will drop down. Then you can scroll up on this pen, and there you can find your latitude and longitude coordinates. Apple Maps, if you have an iPhone, works pretty much the same way, and I'm sure the Android apps are also similar. Another way we will be collecting data is using iNaturalist. iNaturalist is an online social network of naturalists, citizen scientists, and biologists built on the concept of mapping and sharing observations of biodiversity across the globe. It, too, is an app you can download for your phone. It's this little green bird logo. Like other social media type things, you'll need to create an account with a username, another password to memorize, yay. Once you've done that, you can make so-called observations of the species you are collecting using your camera. Just take a picture within the app or upload a picture from your photo file. After uploading a photo, you can select what did you see. You can either enter a name if you don't know what the plant is, or the app will suggest one for you. It will also provide a list of other suggestions if you don't agree with its predictions. The artificial intelligence associated with this app is pretty good at identifying plants through photos, and it's getting better all the time, but it's not 100% accurate. iNaturalist also relies on volunteers to verify identification. After selecting a name, the observation will be uploaded to the site. Your location will automatically be recorded with the observation as the map at the bottom. This is from the iNaturalist website. This is where your observations ultimately will end up. There are a variety of ways to filter the data, which I won't go into now, but we are researching ways to make our work more efficient using them. One of those ways would be to create a project just for tap observations. People from all over the world submit observations to iNaturalist, and I actually look at ones for Oklahoma just for fun because I'm kind of a nerd that way. Also, I've been able to track down sites of rare species using iNaturalist observations, and even this spring, I was able to find species that were new to the state using iNaturalist observations. Having both paper field forms and iNaturalist observations will help us keep track of our voucher and our seed collection. With multiple volunteers all over the state, it's often hard to do that. In this way, we will have multiple pieces of documentation for our collections. With the TAP work, you may be wondering just who exactly is responsible for collecting the voucher specimens and the accompanying data. We are still ironing out the details of the actual workflow, but this is a general idea of how it could work. And this doesn't really pertain to individuals out doing work on their own. This is more about part of the grant. We're working with another individual at the Oklahoma Biological Survey, Priscilla Crawford, who has these sites called Registry Sites. This is more in conjunction with work that will be done at those sites. Just as an aside there. So with those sites, one person who we're referring to as the expert will be collecting the voucher specimens and filling out the associated field data sheet at a particular site. This expert is someone who is already familiar with the species and is relatively confident with their identification skills. This person will be the one to scout out collecting sites when the plants are blooming and they can be positively identified. This is when the voucher specimen will be taken, well before any seed collection is actually done. The expert will fill out the field data sheet at the time the voucher is collected, as well as take an iNaturalist observation. Volunteers later on will visit the sites when the seeds are mature. They will know what species they are looking for based on what the expert collected earlier in the year. They can use the iNaturalist observations made earlier in the year by that expert to get a good idea of what the plant looks like. Volunteers will also make iNaturalist observations of their own from the plant from which they are collecting seeds. This is another way for us to get the seeds correctly and positively identified. And that is actually all I have for today. Collecting voucher specimens and field data are really things that should be demonstrated rather than discussed. So someday soon, I hope we can actually have a field day where we could do all this stuff in person. But we have plenty of time for questions, so I hope you have some good ones. And of course, I would be happy to help you via email at any time in the future as well. So thank you so much for your time today. I appreciate it.
Well, and thank you, Amy. And so we already have questions starting to roll in. The first question we have for you is, how many species are in Oklahoma are there that potentially could be collected?
We have our working number that we use when people ask this question, approximately 2,500, so 2,500 species. Right now, actually over the summer, a few of us have been working on compiling the master list of every single species in Oklahoma. And that number changes. You may be wondering, why doesn't she know the exact number off the top of her head? Well, of course, as I mentioned, we do get new species coming into the state all the time. Unfortunately, a lot of these are exotic weeds, but we do get a few cool new sightings of things that are native to the United States as well. Also, because of the nature of how plants are divided into species, I'm trying to think of a good way to explain this. Names change, and what is related to what changes, it's kind of hard to explain. For instance, there will be a group of plants that's one species, and then somebody decides, no, that's not one species, that's actually two species. So things change all the time. A species is not a static entity. And because of that, our numbers change. I know that's kind of hard to understand. Probably somebody could explain it a lot better than me. But again, let's say 2,500 species in Oklahoma.
Okay. Very good. Okay, next question. Oh my goodness, I have so many questions rolling in here. Do you need mothballs to protect the collection?
Very good question. And obviously somebody who is familiar with collections. No, you don't need mothballs to protect the collections. And in fact, they're carcinogenic and they shouldn't be used. There is a collection down in Chickasha at the college there that has just been saturated in mothballs. Oh, the same thing at Southeastern Oklahoma State University. I bet somebody is familiar with that collection, and that's why they asked that question. The smell of mothballs is so bad, it permeates your entire self, and you smell like that on your trip home, and it's just awful. And while it does work to some extent, you should not do it. You really, really should not do it. We try to keep pests from coming in the first place. And before any specimen can come into our herbarium, we're freezing it at minus 80 degrees for 1 week. Hopefully that kills a lot of stuff before it even gets in. But in the herbarium itself, we just try and keep it really, really clean. We're vigilant about any plant material that enters. And I've been using these plastic pet strips in each individual cabinet. They don't have a smell like the horrible mothballs do, but they seem to be pretty effective too, and not nearly as scary as the mothballs. So while insect infestations are a concern with this sort of collection, most collections are not using mothballs. And Jane, for the collections at PAC, this is something that we will have to discuss.
Right. So we have money for a cabinet, a half cab, and so hopefully that helps a little bit. But we don't have a freezer that goes down to minus 80, so we got to figure out how this do. And we can't use a lot of pesticides, obviously, because of where we are and what we do with a lot of our stuff. So we'll have to figure out how that works.
Yeah, we will figure it out. No problem.
Okay, and okay, well, we'll get more to that in a minute. Okay, next question. You may have mentioned it, but how long do these vouchers last?
These vouchers can last a really, really long time. In our collections at OU, the oldest ones we have are from the late 1800s, and in fact, the labels on them say plants of Indian Territory. So those are sort of interesting. The oldest herbarium collections known are about 450 years old. So they can last for a really, really long time. The paper we use is 100% cotton, and so it doesn't sox. It doesn't get those little orange rusty marks that sometimes old books have. The paper we make our labels out of is also 100% cotton. We sometimes use these little linen strips to stick down heavy, bulky parts of plants that won't really get stuck well with that owner's glue. Those are archival. So in theory, they should last a really, really, really long time. And those in the book that I showed you, well, those are actually the oldest ones at OU. Those are like from 1810. So a really, really long time, as long as you don't have a bug infestation, of course.
Right. And so, Amy, you had mentioned that you were digitalizing, you were scanning in and digitalizing some of your specimen or whatever, the vouchers. We had talked about that a few months back. Can you tell us about that?
Yes. Those images of the voucher specimens you saw, whoops, I was going to go back, but these are actually part of that project. We have money from the National Science Foundation to take high-quality photographs of all of our specimens from Oklahoma and Texas. That's just the way the grant was written. Someday we will have images of all of them. To take photos of all of our specimens. And these are going to be available online. And Oklahoma and Texas are sort of the last in the country to do this. If you look online right now, specimens from states elsewhere are already there. So this is an ongoing project throughout the United States, and it's just a way to make data more available to everyone. In the past, if someone wanted to do research, say, on Baptisia australis, that is the specimen on the left, that's our wild blue indigo, I would have to get all of our specimens, pack them up very, very, very fastidiously, and ship them off. The person there would have to unpack them, use them for the study, and then someday ship them back to me. And as you can imagine, that's a lot of wear and tear on these pretty fragile things. So having images available online is going to help with that. And just like everything else, we should be further along with that work, but because of the virus, we are kind of lagging behind. In fact, I have to do some catch-up scanning all next week, and I'm not really looking forward to that.
So with some of those scans, would those be something that TAP could get color copies of and then share with people?
Oh, yes, of course. And we did talk about that. And in addition to some pictorial guides that we are going to work on, we are going to have images just like this of our target species printed out and put into a little booklet for you guys to use as well as identification guides. Hopefully that will help a lot, in addition to the online pictures through iNaturalist and through the pictorial guides that we hope to get together someday.
Right, and that when we get there, I think is going to be really exciting, because what we've been talking about is being able to have that guide to help people better identify things out in the field, and to have it broken out by seasonality, by month, depending on which region, by season, by color, by height, by location, along with traditional cultural uses, and which pollinators and animals that plant species is important for. So we're really excited about that, and hopefully that'll be moving along sometime fairly soon here. Everything's been a little derailed. Okay, next question we have. Do any of the Oklahoma herbaria have ethnobotany collections?
No, not specifically. We have one collection, and it's not segregated at all. The specimens are just sort of mixed in with everything else from, I believe, the Caddo tribe. I can't remember exactly. I was not involved in that, and it was maybe 25, 30 years ago. I've just seen the labels. So no, unfortunately, we do not have any dedicated ethnobotany collections. And to my knowledge, no one in the state does either. OU is actually the largest collection, obviously followed by Stillwater.
Okay, well, maybe this is something TAP needs to take on, I think. So anyway, another goal, another grant, another goal. Next question we have here, we're going to need a lot of help for that. Which taxonomic key book would you recommend for here in Oklahoma?
Well, right now we have the Flora of Oklahoma. It's specifically titled Flora of Oklahoma, Keys and Descriptions. And it's not a hardcover, nationally published thing. It's a copied and spiral-bound thing that you have to order online, not through Amazon. And of course, if anybody wants that information, please email me and I will get it to them. However, it has no pictures and it is rather technical. So if you've had a plant identification class somewhere in your past, you probably would be able to use it. But if you are a total and complete amateur, it might be sort of off-putting because botany has a lot of specialized terminology. You know, why use a regular word when you could make up a new one? I think that's the motto for a lot of people who describe plants. So there are a lot of sort of difficult words. You wouldn't believe how many words they use to describe various types of hairs. It's incredible. But that is the book that we have for Oklahoma. It's in its second edition, yes. And it's constantly being worked on. And in fact, I am part of that group, and we should be meeting every month, but for obvious reasons, we have not met since last February. So, depending on what part of the state you're in, you can sort of cobble together using other states' books. If you're in the Northeast, the Flora of Missouri would be a good one to get. It's actually in 3 volumes, and it has line drawings of some of the species. If you're in central to south central Oklahoma, the Flora of North Central Texas is really great. It's got line drawings for everything, and it's pretty easy to use. The language isn't nearly as convoluted as in the Flora of Oklahoma. There's Flora of Arkansas that is old. I believe they're working on a new one, however. I use the Flora of Arkansas whenever I'm working in southeastern Oklahoma. And for out west, there is a very old Flora of the Great Plains that people often use as well. If you're very, 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 very far west, like out at Black Mesa, then I've used the Flora of New Mexico even. So there are a variety of options depending on where you are in the state.
Okay, and this is another opportunity to do a pitch at TAP. For our lending library, we do have all sorts of great field identification guides and some basic kind of botanical identification beginners books, and all of those books can be checked out and borrowed for free, so just get in touch with us if you're interested in that. So next question, do you need many specimens from different locations?
Always. Definitely. So we want plants collected all over the state from many points in time because, of course, Oklahoma is so diverse. I mentioned southeastern Oklahoma and Black Mesa. The plants that grow in one of those places most certainly do not grow in the other places. So we want collections from all over the state.
Okay, good. Next question. What are a couple of species the herbarium is having difficulties finding?
Oh, my goodness. So Oklahoma kind of sits in the middle of everything. We're on the edge of the eastern deciduous forest and well out, of course, at Black Mesa. You're at the foothills of the Rockies. And kind of the center, it's all the Great Plains. Basically, Oklahoma is kind of a crossroads where a lot of different ecosystems come together. Consequently, we're at the edge of the range of many species. So we consequently have a lot of things that are rare to us. You go over the border and they're pretty common, but they're rare for us. So some of the things I've been involved in looking for over the years, because of the work I do with the Oklahoma Natural Heritage Inventory, which is a program that tracks rare species. The Kentucky lady slipper orchid, we've been looking for those always. I think maybe there are about 12 stems total in Oklahoma. Out in the Panhandle, we've been looking for the wheel milkweed. Of course, TAP is very interested in milkweeds because of their pollinator value. This is one that I've never seen. That's only been seen maybe twice in the state. It's about 2 inches tall and has triple flowers. Super cute. I'd really like to find it. Oklahoma actually only has one endangered plant species, believe it or not, and that grows down in southeastern Oklahoma on the lower mountain fork. We've been all over that area looking for other populations, but we haven't been able to find them. So that's another example.
Okay, what is that what's the name of the species here in Oklahoma that it's endangered?
Oh, that species is called Herbarella. The scientific name is Herbarella nodosa, and it is probably the most unexciting looking plant you could possibly imagine. If you're familiar with how the flowers on carrots or parsley look, they form that sort of umbrella, upside down shaped umbrella inflorescence and have sort of lacy leaves. It's a member of that family, and so it kind of looks like that. It's maybe 3 inches tall. It's aquatic, or semi-aquatic, meaning it's mostly underwater a lot of the time, and it looks like absolutely nothing to most people at all. It's not very exciting, unfortunately, unless you're a botanist, and then it's very exciting.
Okay. Well, very good. So I think that's our end of our questions. If you've got any more questions, it's not too late. You can go ahead and type them in the little chat box there. But I will take this opportunity to give another pitch for if you're interested in what you've heard today, if you've enjoyed this presentation, want to take this to the next level, get involved in this effort, please, we need your help. TAP needs your help. The butterflies and the pollinators need your help. We all need your help to try and build this seed bank. It is the only one of its kind in Oklahoma. I think it may be in the region. And we're trying to build it and have more robust biodiversity on our lands by restoring them with these plants. So next year, when things get back online, we had to pause this year, as everybody did, but we are making plans to resume in-person training next year. It'll probably be outside at the farm, but we're kind of gearing up for that, to switch our education efforts to being pretty much 100% outdoors. But we're going to be doing field ID training. We're going to be doing hands-on training with making these vouchers. The whole thing, and then also we'll have access to some new collection sites on these on the registry with the natural areas. So we're going to be doing field identification training so people are ready to go out and do those kind of things. And anyway, so it's pretty exciting, but we need help with this effort. And again, if you liked what you heard about today, we're going to be doing a lot more in depth of these kind of things moving forward, and we would love your participation at TAP. So with that, I think we're done for today. No more questions are coming in. But Amy, thank you so much. That was really fascinating. It got me very geared up and wishing that I was out there in the field doing this and collecting and learning about all this. So thank you, and we look forward to working with you and all the folks over there at OU as we continue to move forward with this project.
Thank you, Jane. I appreciate it.
Okay, and thank you for your time. It was really, really informative, and thanks for all you do. And everybody, thank you for tuning in today, and we'll be having another webinar soon. And anyway, stay safe, and we'll check in with you later. Thank you. Bye-bye.
Bye-bye.